This Week in Virology, the podcast about viruses, the kind that make you sick.
From Microbe TV, this is TWIV, This Week in Virology, episode 563, recorded on August 30th, 2019. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. It's beautiful late summer day. It's up to 85 today, but getting lows in the mid-50s at night and having the windows open. Just lovely weather.
23 Celsius and sunny. 29 Celsius, pardon me.
That's what we've got here.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, good to be here. It is just as beautiful here, and I'm really looking forward to enjoying that nice weekend.
Mm-hmm. Then you have to start teaching next week, right?
I started teaching this week, so today was my last day of week one.
So Princeton, where I was this week, doesn't start for another 2 weeks.
Wow.
They have some weird semester, maybe quads or tri-semester.
Do they have quarters or
quarters? Maybe.
Trimesters?
Yeah, and they're changing, I think, in 2 years.
Okay.
They're going to be in line with everyone else. Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. Here it's also lovely. 24 degrees Celsius, 75 Fahrenheit. Blue sky, no rain.
Nice.
You were soon to say something, Brianne.
Oh, I was only going to say that was a little strange for Princeton to be starting so late. They wouldn't have gotten to learn about complement today.
Yeah, right. Yeah, I was there, and it was very calm. Without students, Princeton is very calm. With them, it is very hectic. But we were walking to lunch one day, and we passed the president of the university. How about that?
Cool.
So if you like what we do here on TWIV and the other podcasts, consider supporting us. Microbe.tv/contribute. Someone said, you should tell everyone that you do all this by yourselves. Yeah, we do. All of us. So we all record, and then I produce it myself. I edit it and post it. There's nobody helping, with the exception of TWIM, where Ray Ortega is the producer. He edits and posts. I do everything else after the recording. So if you'd like us to get some help, you can microbe.tv/contribute. If you don't want to get me some help, that's fine, but still contribute because we have expenses and we would like to go places a little more than we do. We were just at Rutgers a couple of weeks ago, Brianne and Dixon.
We were. It was great. It made me very excited to be a person in a broad biology department.
Are you saying that this is not a broad biology department?
No, it was.
But you're right.
Nice
you are.
to get to think. Well, I was saying that it was just really nice to use all of the different types of biology that I talk about with my colleagues so often.
No, you're absolutely right. Like here at this medical school, we do not do broad biology. We do mostly, human disease related science. And I think it's really cool to learn about diatoms. But we don't have diatoms. We don't study them here at Columbia Medical.
I told my marine ecology colleague all about everything we learned about diatoms and coccolithophores, and she was excited to write them into her syllabus.
Cool.
So I spent this past week was at Princeton working on the textbook, but the previous week I was in Sweden having a very nice time at a meeting and doing 3 different podcasts. So you'll hear those in the coming weeks. A lot of fun. And I met some really cool people there, I have to say. So Kim Vesto is a PhD student at the Karolinska. He arranged one of the TWIVs. And Agnes Wiener is at the Karolinska. She arranged one of the others. But I have to say Agnes was amazing. She's a clinical microbiologist at the Karolinska. I had met her in Rotterdam. And she said, if you ever come to Sweden, let me know. So I let her know. And she did so much. She picked me up at the airport. She drove me everywhere. She brought me to the Nobel Museum, the the dining hall at City Hall where they have the Nobel banquet, a whole bunch of other stuff. And then she drove me and Erling to the meeting, which is a few hundred kilometers on the other side of the country. But on the way, she stopped at all these cool places that she knew about. It was just amazing.
Oh, cool.
Nice. Yeah.
Nice.
Including, and in Stockholm, of course, she brought me to the Vasa.
Yes.
And in fact, she had emailed, she said, since you guys talked about the Vasa on Twiv, you should come see it. And so we got a little tour, and the guy said, someone asked him, why didn't it rot? And he said, because it's brackish water and there are no worms. Right? So Dixon had said it was fresh water.
Yeah.
And someone wrote in and said it.
No,
You did. That's right. You did. So, and it's an amazing thing that they put this together, right, Kathy? It's huge. And you can walk
it's huge.
right next to it. And they have a little movie there that shows you how they did it. It's just stunning. And you said you spent 5 hours there, right?
Yeah. And I had seen it once before. I had seen it in 1985 also. So yeah, it's even bigger and more stuff to see now than then. So yeah. And
the museum they built around it is pretty cool.
Yeah.
They have a lot of exhibits. So I highly recommend the Vasa.
Yeah.
And then, then I went to Smögen for the meeting, which was, and Agnes came to the meeting as well. And then she drove me back. So this is normally like a 6-hour drive from Smögen to Stockholm, but she wanted to show me all these things. So it took us 12 hours to get back.
Wow.
But on the way, so one thing we saw were these cool Bronze Age carvings in rocks.
Ooh.
Where they drew pictures, and they drew a lot of ships, but they also drew people. And it's pretty cool. And then there are lots of locks where they move boats around canals.
Mm-hmm.
And there's this one place where they built a bridge for the boats to go over a canyon.
Oh.
And the bridge is
I think I've seen pictures of this.
It's amazing. And the bridge is flooded with water, right? So,
right.
So a boat actually went across it while we were watching it. And I'm like, my gosh, how did they have to engineer this so it was strong enough to hold the boat, right?
Yeah.
And the water.
Right.
And the water is very heavy as well. It was really cool. And then finally, she stopped at this little town where Picasso had installed a huge outdoor sculpture in the middle of nowhere. It was right on the coast. It was really beautiful. So thank you, Agnes. It was great. And I have to go back because the country is so cool and there's so much we didn't do. But more because Agnes will show me around. So thank you so much. It was really, and then many other people I met. Thank you all for being really nice people at the meeting. Were great. So you must know. Did you know Erling Norby, Kathy? Because he did some ADNO work, right?
I know the name, and I can't remember if I've ever seen him or met him.
Yeah, he's an interesting guy. So we had a nice conversation at the meeting about Nobel Prizes in science. And since we didn't have enough time to get through more than 4 of the questions I had planned, he's actually visiting New York in October, and he's going to come and we'll do some more.
Cool.
So I had a lot of fun. Thank you, Sweden. Thank you, everybody. And you and Kathy was in Norway. That was cool, right?
Yes, I did. And my pick will reflect that in just a small little measure. But yeah.
Is that your first time to Norway?
No, I had been to Oslo in 1985. So same trip as I saw the Vasa in Sweden.
Yeah. So they said that I should go to Norway. It's really interesting. But they said if you have a choice, you should come back to Sweden. I probably shouldn't say this. No, I won't say it. Another country, they said, don't go.
Ah.
Uh-oh.
And I learned something that I hadn't really figured out the distinction of before, but Scandinavia includes Norway, Sweden, and Denmark. But when you say Nordic countries, then you can also include Finland and Iceland.
Oh.
But evidently, Americans kind of conflate all of them into being Scandinavia, but that's not true for Finland and Iceland, technically, if I understood the person who was telling me. Yeah.
Well, I'm off to Copenhagen in 2 weeks, so I'm going to Scandinavia again.
There you go.
I like Copenhagen. It's really fun.
Never been. First time. First time to Sweden also, so looking forward to doing that. So we have some follow-up from the last couple of episodes. The first one is from Anthony about our Rutgers episode. I couldn't find a "Have you hugged your phytoplankton today" bumper sticker. Have you thanked a diatom today bumper stickers I did see in 2 designs. And he gives a link to a Zazzle, because we talked about how you should be grateful for phytoplankton and diatoms every time you take a breath.
Right.
Which is why the episode is called Take a Breath. It all too very often seems that New Jersey isn't a state, but a punchline instead. That's pretty true, right?
Yes, it's very true.
Indeed. I didn't think that there was much of anything to be proud of here, with the exception of Merck making possible the hope of the eradication of river blindness. That was before TWIV 562. Thanks go to you and your colleagues for putting New Jersey on the map in a good way. And of course, Selman Waxman and antibiotics, right?
Yes.
Which I think we must have
saw
mentioned.
the Waxman Museum.
It was very cool. And a lot of good stuff going on there. And mostly, we talked about non-human viruses, but there are also people at Rutgers doing human virology as well.
Mm-hmm.
So that was very cool. Kathy, can you take the next one?
Sure. Igor writes, Dear Vincent and the gang, I imagine thousands of people from Regeneron emailed you about the way antibodies to Ebola were derived at the company. But just in case the number is a bit lower, here's the link to the paper describing the VelocImmune mice. He gives the link. The mice are genetically modified to replace the mouse immunoglobulin heavy and kappa light variable region with that of the humans. All antibodies in these mice carry fully human FAB regions and mouse FC region, so it's very easy to humanize them after isolation. Also, here's my thoughts as to why the term quasi-species is getting replaced with swarm. Quasi-species were introduced by Eigen back in the 70s to describe early evolution of life on Earth, and then later expanded to viruses by Domingo, Holland, and others. However, originally the term was quite specific. In fact, it was defined in mathematical terms in population genetics models and was used to describe a situation when a collection of closely related clones has a different biological behavior than any single clone. It was argued that in such cases, in evolutionary terms, the quasi-species serves as the unit of selection, not individual clones. Later, virologists started using the term quasi-species to refer to any collection of closely related viruses, especially to the rapidly mutating RNA viruses, which often exist as swarms of mutants. The old guard was not happy and often corrected the incorrect use of the term. Just to give one example, in this article by Wilkie, he writes, virologists in general have frequently used the term quasi-species inappropriately. I guess the pedants won, and folks began to use swarm rather than quasi-species to avoid hearing for the 100th time that they are using it wrong. Thank you for the excellent podcast. I listen to you every week on my long commute to work. Best regards, Igor.
Now, I'm wondering when the entomologists are going to start complaining about our misuse of the term swarm.
Yeah, that's right.
Because most
Probably
swarms are clonal. They're all descended from the same mother, so I don't know.
But they might object to our use of the term bugs before that. So,
well, yes.
Hey, some of us sometimes, not us here on the podcast, but some of our colleagues often use the word bugs.
Sure.
Right?
Yeah.
To describe microbes. So,
exactly.
Right, 2 things here. This paper is very cool. These VelocImmune mice, which, we didn't know about because we hadn't. Actually, the thing was meant to be a news report, a brief news report, and then we got into the science.
And then I started asking questions.
And then we didn't realize we hadn't researched it. But these mice, as he said, they have, all you have to do is replace the constant region of the antibodies after you make them. So what's incredible is that they knocked out the locus and then replaced it at another site. And you can immunize these mice with whatever you want and make monoclonals. Mm-hmm. So they go through rearrangement and all of that to get diversity. It's pretty cool.
Yeah, it's fantastic.
So here's a quote from the paper. The immediate utility of VelocImmune mice is evident by the more than 10 different fully human antibody therapeutics that have already been produced and entered into clinical development in a short period. In addition, these mice are perhaps the most valuable ever engineered, already having brought in more than $2 billion in committed licensing fees and collaboration revenues.
Clever girl.
Wow.
Those are the words of the authors of the paper at Regeneron, who are clearly very happy about these mice. But of course, they make very nice monoclonals, and 3 of those are the ones being used in that Ebola therapeutic. And by the way, since we mentioned that, someone over on Twitter said, you guys were so enthusiastic about those monoclonals. Why don't you consider it from a public health perspective instead? I don't know. I thought we were sufficiently circumspect by saying they had to be, given intravenously, which is, and so forth.
Right.
I don't know what else they had in mind.
Yeah, I'm not sure what we missed from a public health perspective.
I didn't, I said, I thought we did it, but then there was no follow-up. So if anyone knows why we should not be enthusiastic, I doubt any
Please
of
let
you.
us know.
I doubt any of you are at Regeneron, right? The other part of this email, the quasi-species and swarm. So the article is actually very interesting. It's open access, so you could go check it out. And here, I'm going to just read you a little bit. These arguments do not imply that quasi-species theory is the final answer to all questions of virus evolution. It has shortcomings that need to be addressed. So he says basically it's okay to use either term. I understand that Eigen originally had it in mind for very large populations, and I think virologists tend to not use it that way. But he says it's okay. It's not really an issue to use it.
And language is dynamic.
I love quasi-species, even though some people don't. It seems it's a great word, right?
I think as long as we have a clear idea of and an agreement on what it means, then it should be fine to use, right?
Mm-hmm.
I guess it's
if
just that,
people are using it to mean different things in different settings, then we would need to address that. And
right. And I think of reverse genetics along those same lines as meaning different things in different settings, and therefore, yeah.
So it says in the abstract, a number of recent papers have cast doubt on the applicability of quasi-species concept to virus evolution and have argued that population genetics is a more appropriate framework. And he concludes, since quasi-species theory and mutation selection balance are two sides of the same metal, discussion about which is more appropriate to describe virus evolution is moot. And he said, in the future, we should focus on the important questions. So, yes, I think that's a good conclusion there. Not worrying about it. So, Igor, I think you've moved. As far as I remember, you used to be in New York City. And now you're in Worcester. So, well, good luck there.
yeah.
In any case, he makes good points.
It did.
Yes.
Very good. Excellent. Thank you. Alan, can you take that last one?
Sure. Christos writes, Hi, Vince. I hope everything's going well. I'm listening to your most recent podcast episode. In your search for the Regeneron monoclonal antibody combo, you found the clinical paper, but you missed the preclinical paper. I'm attaching it here. We are describing the development properties and most animal studies with these antibodies, and provides a link.
Yes, it is development of clinical stage monoclonals. And they describe using the VelocImmune mice, which we had missed. And they selected 3 that bound to different epitopes. And listen to this. How did they determine the epitopes? Binding and electron microscopy. Just look and see where they bind.
All right.
It's very cool. And then they tested them in non-human primates to make sure that they prevented infection. So we had one person from Regeneron, not the thousands predicted by Igor, but that's fine. And Christos is clearly listening. I wondered if he would pick it up. I have to tell you that Christos was a graduate student here at Columbia with Saul Silverstein. I was wondering
if this is Christos Panagiotis, right?
No, Kyriatsus. It's another Christos. You know Christos Panagiotis, yeah.
Oh, okay.
Now, this came after your time. But I was on his thesis committee. Okay. Now, Alan had sent us, here we go, over on, so we started using Slack. Thank you, Kartik. And you had gotten a tweet from Reg Reader whether we should be getting a flu shot in August.
Right. So the topic that came up was, I guess, she'd had an opportunity to get a flu shot and wondered if that was a good idea. And I thought this is a topic that's kind of complicated and crunchy enough. We probably ought to address it on TWIV.
So that's our snippet.
Yeah.
It's crunchy and complicated, and we're not going to resolve it.
We're not going to answer it.
We're not.
But here's some
I
information.
learned a lot from the paper, though.
Yeah.
Mm-hmm.
So the paper is Clinical Infectious Diseases, and it is published in 2017. Intra-season waning of influenza vaccine protection. Evidence from the U.S. Influenza Vaccine Effectiveness Network, 2011 to 2012 through 2014 to 2015. And this comes out of the CDC and the University of Michigan School of Public Health and Marshfield Clinic Research Center in Wisconsin, also in another research institute in Seattle. So this is a question that people look at, asking how effective is the flu vaccine? And so, you can start getting it now here in the Northeast in the U.S., I guess, right?
Mm-hmm.
Is that too early?
Yeah, is it too early? And, the flu season typically starts in December or early January, but it can vary. And there's been arguments that if you get it in August or even the end of July, that by the time the flu season comes along, it's the protection wanes, right? Especially if you're older.
Right.
And I typically get it in October, November, only because that's when I get around to it.
Mm-hmm.
But all right, so there is a influenza vaccine effectiveness network in the U.S. And what they did in this study is to enroll adults and children who were looking for outpatient care for respiratory illness, and they had 5 sites in the U.S., Seattle, Marshfield, Wisconsin, Michigan, Texas, and Pittsburgh, Pennsylvania. And the infection is confirmed by PCR using CDC-approved methods. And the subjects are considered vaccinated. This is an important part of the study because you want to know if these people who are sick and have confirmed flu, if they're vaccinated or not. So they have to have documented evidence in their medical records of 1 or more doses, at least 14 days before illness onset, because that would be how long it takes to get a response to the vaccine. And they looked at a lot of people in this study. So the times are January 2014-15, that's the onset, and they also looked at 2011, 2012. 11,000 observations of H3N2, 4,100 for H1N1, and 5,500 for influenza B virus infections, because the vaccine contains components of all of those viruses. And so then they can take all this data and they apply various algorithms to look at it. And they make the following conclusions. So it's called vaccine effectiveness, or VE. The VE against H3N2 declined with time since vaccination, and the maximum effectiveness is 35% 14 days after vaccination. 158 days later, it's 0. 0. I actually don't want to say this because now a lot of people are not going to get vaccinated.
Yeah, and
there's a point in the discussion that we can bring up later.
Yeah.
This is all from mathematical modeling.
Yes.
0 is not 0 here. Yeah.
Yeah.
7% decline every 30 days in vaccine effectiveness.
Right. So the 0 is an extrapolation.
Right.
Exactly.
Because they didn't look for that long. They look when the people come in, right?
Right.
Mm-hmm.
They're coming in at different times after vaccination. They have to make an extrapolation. H1N1, maximum vaccine effectiveness, 80%, which is 14 days, and then 37% 120 days, 28 days later.
And still 46% at 180 days.
Yes.
6 months later. So quite a bit better. Yeah.
6 to 11% decline every 30 days. And then influenza B, maximum VE 59% at 14 days, minimum 23% 180 days. It declines 7% every month. That's the results of this study, but there have been many others. Right?
Yeah.
Mm-hmm.
They say our findings are similar to other reports which suggest waning effectiveness for H3N2 in Europe. Oh, okay. So here's something that's very interesting, worth talking about. The decline of VE is more pronounced in subjects with prior season flu vaccination. And
Yes.
and I would suggest that repeat vaccination may blunt the response to the vaccine. And that's a complicated issue because there is this phenomena of, original antigenic sin, where
Yes, that's the one.
you usually make a response no matter what you're infected with to the first strain you ever saw. So that can complicate matters.
There's also there's a confounding factor here. People who get a flu vaccine every year is a population that's going to be enriched for people who are more susceptible to flu.
Right.
So, and I know they've corrected for things like the obvious stuff like age here, but you may be looking at an effect that's not actually due to the vaccine there.
Mm-hmm. Yeah.
Yeah. They do a nice job talking about that and sort of some of these limitations of the selective populations you may be seeing here. The thing that I found really interesting in the discussion is they pointed out that one of the other studies had been different in that it actually looked at antibody levels in people over time. Instead of doing the mathematical models, they looked at antibodies, and they found that the antibodies, it took more than 58 weeks to decrease twofold, which is pretty different from what they see in the mathematical models in this paper.
Mm-hmm.
And they indicate that might be because their models can't account for the fact that flu mutates during the season.
Right.
And so maybe those people who got the early vaccine are being infected with a mutant form of the flu later in the season.
Yeah. Yeah.
So the vaccine might have lasted, but the flu might have changed.
Right.
Yeah. They say that serology is consistent with a modest rather than a sharp decline in protection within a given season.
I did appreciate that they used multiple years of data here. A lot of studies on vaccine effectiveness look at a single year, and there are arguments both ways. There's an old saying in the flu epidemiology community that if you've seen one flu season, you've seen one flu season. Because it's, it may start in September, or it may start in February, or, and it may be bad, or it may be mild, and you get different strains. And so this, where they're doing, I guess it's a 4 or 5 year slice, they're actually hopefully averaging out a lot of that variability that you would normally see. Of course, the downside of that is that you've averaged out signals that might have been useful to look at. So as I say, there are arguments both ways.
Mm-hmm.
Yeah, I was just going to say that one of the paragraphs in the discussion, they talk about it's possible that the observed pattern of decreasing vaccine protection arose from uncontrolled confounding.
Yes.
Yes.
It's a phrase I hadn't seen before.
I highlighted that.
Yeah. And they also talked about using spline functions. That's the mathematical modeling that they used. And I had never heard of, I can't find the specific phrase here now, but it's some kind of spline. It was a 3-word phrase, and I Googled that and was not particularly enlightened by what I found. But that was also new to me.
Natural cubic spline.
That was it. Yes.
Yes.
So, yeah, there's a whole world of mathematics out there that I did
not dig into the mathematical modeling because that's a deeper rabbit hole than I can get into, I think. But
at least get out of.
Get out of, yes.
I think an important point here is that this is not a clinical trial, which is what you would ideally want to measure vaccine effectiveness and correlate with the time of vaccination, right?
Yeah, this is an observational study.
It's observation. They make models to extrapolate, but that's not perfect. But they say, how would you design the trial? Because when you think about it, you'd need to have people who get vaccinated in July, in August, September.
Right.
And then you'd have to follow them through the season. You can't just do a big population study. So it would be hard to do.
And you'd have to have enough people that some of them would actually develop flu.
Yep.
Right.
Yep.
Yeah.
So it's a hard study, and it probably would cost a lot of money, and it's not clear that anyone is going to do it. So that's the limitation. So this has a limitation. They also point out there is little evidence that large numbers of people in the U.S. are getting vaccinated considerably earlier than has previously occurred. That's very interesting because I see the signs out already. Get your flu vaccine, right?
Mm-hmm.
Of course, I also see the Halloween candy on display at the supermarket. And it's still frickin' August, but
Yeah.
Anyway, I don't know what to tell you. I think you can do what you want, right?
Here's the way I look at it. Here are the facts that they say in 3 seasons, the influenza season started by the beginning of December, and in 3 seasons, it started in or later than the 3rd week of January. Okay. Duration of the annual epidemic averaged about 13 weeks, with some seasons lasting as long as 17 weeks. So 4 months plus a week. And if the vaccine takes 2 weeks to become effective, and the season usually starts by the beginning of September, that would tell you, November would be good enough.
I think so.
But we're not that kind of doctor, and we're not making that recommendation, but those are the facts.
Well, and another something to consider here is if you see the sign outside the pharmacy in August, get your flu shot, and you say, hey, I've got 10 minutes. I'm going to go get my flu shot. Then you've gotten your flu shot for the season. And if that contrasts with maybe back to school time and you get busy and you don't get around to it and it gets to be December and it's the middle of flu season and you still haven't gotten it. I mean, if this is a choice between getting it and not getting it because it was more convenient to get it in August, then get it in August.
Yeah.
But I personally tend to wait until the flu clinic that our town does, which is usually in October, just because that's convenient for me. But if they decided to start having their flu clinics in August, I'd probably go to it in August.
Here's how I decide. Dixon comes in and says, why don't we go get our flu shots?
Yeah, that's good.
And that's usually October, November.
Yeah.
And I put it off a few weeks. And then he says, I'm going on my own. He says, okay, I'll go. Because we get it right here in the hospital. Mm-hmm. But there you go, Reg Reader. Your mileage may vary.
Yes. But
who knows if that was actually helpful?
I don't know if that helped at all, but do get your flu shot. And I think if you have your choice of times to get it, autumn is probably a optimum. I mean, as Kathy was saying, based on what this study says and based on what we see from previous flu seasons, October, November is an excellent time frame, and you're likely to have lots of immunity whenever the flu season hits after that.
Yeah. I felt like it was October when I read the study, and then any time when I read the results about the antibodies.
Right.
Has anyone gotten a flu vaccine this year yet?
Not yet.
Nobody?
No, not yet.
I haven't either. I saw the signs, but I said, not yet. I'm waiting till a bit later. Okay. Thank you for our snippet. Now we have a paper for you, which just came out in Nature Biotechnology. It's called Precision Mouse Models with Expanded Tropism for Human Pathogens. And we have a first author, Angela Wall, and we have 2 co-second authors.
Co-second authors, yes.
Have we seen that before? Probably.
Oh, I think so. I think we've seen all the permutations.
All the permutations. Yes. We've certainly seen co-PIs or whatever.
Mm-hmm. Co-last authors.
Chandrav De and Maria Abad Fernandez are the co-second authors. Eric Lenarsik. Now listen, this name sounds familiar. I think that's a listener who has written in to TWIV. It just seems familiar. And then the last author is J. Victor Garcia. So this comes out of a variety of places at the University of North Carolina, University of Oklahoma Health Sciences Center.
And North Carolina State.
Oh dear, I just ignored it because it was North Carolina.
No, if you're mentioning UNC, you better mention NC State there.
Yeah.
Yeah, and NC State, there you go.
Especially since the two of them collaborating on a project is a big deal.
So this caught my eye because it's a cool story. Everyone knows who listens to TWIV that mouse models are important for studying viral infections, infections with other pathogens, diseases of various sorts, but they're not always the best that you can do. Not every virus replicates in mice, and they don't often duplicate the disease, the immune responses, and so forth that you see in people. And so this is a way to address that, which is pretty cool. Now, perhaps we've talked before about humanized mice, for example, BLT mice, which is not bacon, lettuce, and tomato, but it's bone marrow, liver, and thymus humanized mice. You give immunodeficient mice a bone marrow transplant, and you give them pieces of human thymus and liver tissue, and that reconstitutes them with human innate immune cells like monocytes, macrophages, dendritic cells, natural killer cells, and of course B and T cells.
So basically you've taken a mouse that's had its immune system deleted genetically, and you've installed a human immune system in it, is the essence of the BLT mouse.
Exactly.
Yes. It's a very good mouse, but of course, not all viruses are going to replicate in these cells.
Right.
Yes,
right.
It is
That's
still
fine.
A mouse.
It is still a mouse, but it's very useful for studying viruses that'll replicate in those lineages, the B cells, the T cells, the whatever. But it's not going to get you realistic results. It's not going to work for viruses that replicate in lung, for example.
So in these such BLT mice, I suppose, can be used to study HIV infection,
Sure.
right? Because you've got an immune system.
They can.
Yeah.
And they can also be used to study T cell responses because you can actually look at the right epitopes.
Yeah, yeah. So here they say, can we introduce other tissues into mice? And they try it with lung tissue. And so they take a little bit of lung tissue and implant it into the backs of immunodeficient mice.
Human lung tissue.
And these are called LOM mice, lung-only mice, right?
For reasons that'll be clear in a moment.
Now, I just want to take a moment to tell you where this tissue came from, because in the paper they say 2 small pieces of human lung tissue were implanted, and it doesn't say
yeah.
Doesn't say where.
So I searched very hard, and I finally found it in the methods, which is separate. But I was looking in the wrong place originally. I was looking in the supplemental. But there's a separate method. So they say LOM mice were constructed by implanting 2 pieces of human fetal lung tissue. All right. And then in parens, advanced bioscience resources. If you Google that, you try and go to the website, it's shut down because, of course, the president of the U.S. has imposed a ban on the use of fetal tissue in research. So you can't do this experiment, I guess, in the U.S.
So this is an excellent example of important research that is stopped by that change.
So anyway, back to the implantation. You put these pieces of tissues in the mice, and they say over time it expands, it grows, and you get the formation.
But it does not breathe.
It does not breathe. That's right. It's not ventilated. And of course, it doesn't look anything. Don't think that you got a nice trachea with 2 lungs growing here. No, that's not what's happening.
It's a little slice of tissue.
Yeah, it's 3-dimensional, right? It's like not a sphere, but it's what would you say, like a bean or something like that,
right?
And it's got blood vessels. It's got cells of different
bronchioles.
sorts.
It has the substructure you'd see in lung tissue. It's just not shaped like a lung.
It's got an airway epithelium that's ciliated. It's got cartilage and non-cartilage bronchiolar airways, right? So it's the same kind of.
So it's not breathing, but it looks like it could.
It's got goblet epithelial cells, which make mucus. And if you take these cells out and put them in culture and you stimulate them with like lipopolysaccharides, they make cytokines and chemokines, and they're human. This is pretty cool. This thing is growing because it's not been rejected from the mice because they're immunodeficient, right? And that keeps growing, apparently up to 2 months, right?
Yep.
Club cells, which make tryptases that cleave the HA of influenza viruses. And so this is a well-defined, vascularized, and accessible human lung implant because it's right there in the back of the mouse. So you can inject things into it, and it's all human cells.
Yep.
So if you shine a light on this mouse, the shadow will be human. I wrote a blog post yesterday, and I searched for an image, and that's what I found. I searched for humanized mice, and one of the images was a shadow of a mouse on the wall. It was a human. So I used that. So the first thing they do is ask, can we infect this tissue with a variety of pathogens, viruses, and one bacterium? So they inject directly into the implant, and they use first MERS coronavirus, which we know replicates in the lung, but it doesn't replicate in mice. But here we now have human lung tissue in mice, and it does replicate. They do a plaque assay, and they can find 3 log increase in virus titer than the inoculum, and they can show antigens in these cells by staining them. So it's replicating. Then they try Zika virus. They inoculate Zika virus into there, and now here they don't do a plaque assay. I don't know why, because you can do plaque assays with Zika virus. So, they use PCR and measure copies of RNA, and it goes up. But of course, that doesn't mean that you're making infectious virus. Although, did they stain? No, they didn't stain for Zika antigens. So there, you could do a plaque assay. Maybe this suggests that another lab did that. I don't know.
They actually tell in the back who did
what?
All the different experiments with respect to who did the Zika, who did the HCMV, and so forth. I found that really interesting detail.
So PCR is not infectious virus, as we've said before. And by the way, in Sweden, everybody agreed with me. It's probably because they want to be nice to visitors.
Right.
Then they put the tuberculosis bacillus, Mycobacterium bovis. It's actually the vaccine strain. I guess they don't want to work with the actual bacterium. And that's
it's enough paperwork that they're working with fetal tissue to add on the
more paperwork
with live TB.
Yeah. And so that grows in there because of course they can measure colony forming units there, and that's good. And then respiratory syncytial virus, where they have a modified one that has green fluorescent protein gene incorporated into the genome, so they can see it green cells in the lung implants, and they see morphological changes in the cells that are consistent with what we know about viral effects. Human cytomegalovirus, and this is one they're going to come back to in this paper. They measure DNA copies. And they can see maintenance of the DNA 14 and 52 days after inoculation. So, and we know that HCMV establishes persistent lifelong infection. So that's consistent with that. They do an effect where they treat the mice with an antiviral, ganciclovir, which is used to treat HCMV in humans, and they can see that it does reduce viral replication. And so they say that
this is a really nice general purpose viral bacterial lung infection model.
Yeah, it's pretty good.
It was. As soon as I read the abstract, I immediately sent it to my sister, who is doing a lung infection model for her PhD.
Cool.
Well, I don't know if you can do it. Can't get fetal tissue. Can't get fetal tissue.
Oh, can't get fetal tissue. Yeah.
Unless you got some banked, but I don't think you can bank it in a viable way. Anyway, this is great if you want to look at what's going on in the human lung, right? But as we know, when viruses infect the lung, they often go elsewhere and infect other tissues. So you can't do that here. But this is better than nothing. So I agree.
And also, what we're about to get to is that these mice are not mounting an immune response.
Right.
These are immunodeficient mice, and there's a human lung in there. And of course, in a normal human lung infection, you'd mount an immune response, which is a critical part of the whole viral environment.
Right. So they are suggesting that you could use this not only to study viral pathogenesis, but also to look for testing antivirals, right?
Sure.
Because they show the proof of concept with the ganciclovir. The HCMV in people, as I said, establishes these lifelong infections, but you can also see lytic replication in people, and they find that in these animals as well. And that's something that's hard to study otherwise, so they're able to demonstrate lytic replication in this mouse model. All right, so what about an immune response? So now here's where we go back to BLT.
Yep.
They take and they put now the lung implants into BLT mice. So these are now, again, mice with a humanized immune system. Innate and adaptive arms are all humanized. And they can put the human lung into that, and now you get, what is it, BLT-LOM mice?
Yes.
Except they just call them BLTLs.
The BLTL.
Because LOM was lung-only mice.
Right. BLTL. I'm just getting hungrier as we talk about this. Probably because I haven't had lunch yet. And so they can then show that these mice have, all the human immune cells, innate and adaptive immune cells. They make cytokines and so forth. And they can infect them with cytomegalovirus and see what's going on. So they do a lot of characterization of these mice to make sure the immune system is intact, and that's fine. You can find, by the way, these cells, these human immune cells, are all throughout the mouse and in the lung implant as well as on other mouse tissues. Right? Is that accurate?
Yep. Mm-hmm. Yeah. I think so.
Mm-hmm. So then they infect them with HCMV. Now, if you only have the LOM mice, the virus replicates a long time. But if you've got the human immune system in there, the BLTL mice, this titers begin to decline around 11 days, which is, of course, suggesting that it's being controlled by the human immune system. And they can show that these mice are making cytokines and chemokines, and they're making IgM against the virus. They're making T cells, CD8 positive T cells, and they can, determine what epitopes are being recognized, and that's consistent with what we know about the HCMV T cell epitopes.
And this is a lot like what you see in a human CMV infection, right? Mm-hmm. I mean, this is a virus that a huge swath of what's the percentage? It's like most people have had this. And the virus goes and it replicates and it's harmless, and your immune system tamps it down, and there you go. Yeah. You're CMV positive. So the LOM mouse, the first mouse model, develops a much more protracted infection that's not a great model for human infection. This BLTL mouse mounts an immune response and seems to tamp down the infection. So it's a much more realistic representation of how that virus develops.
And the BLTL mice control the infection. They also make IgG, by the way, not only IgM, IgG, so they can do class switching. Yeah. They make CD4 positive T cells after multiple inoculations with the virus. So looks like all the parameters that we know of, or some of them anyway, are they make sense. You infect these, you get an immune response against the virus that you think is reasonable.
And there was an interesting,
like a great model.
Seems so.
Yeah. And there was an interesting additional thing that they did. They mentioned this I think it's only in supplementary data. They had this cohort of mice, BLTL mice, that have much less diversity in their class 1 and 2 alleles.
Yeah, yeah.
So they're homozygous across all their class 1 and class 2 MHC alleles. And those mice mount an IgM response, but it doesn't evolve into an IgG response, and they don't develop any CD8 positive response, which suggests that you need a more diverse response in order to get the full spectrum of immune activation against HCMV.
Right.
Very cool.
Mm-hmm.
Yeah.
So we put a human piece of lung into humanized immune cell mice, system mice, and we can infect them. We get immune responses to the virus.
Against a human virus.
Including within the lung. So this is a nice platform for studying infection, not just viruses, of course, but other pathogens that infect the lung. You can look at what the virus does to the lung. Now, for us, we work on enterovirus 68, where there's no good model yet for studying the virus. We don't even know what cells it replicates in the respiratory tract. This would be perfect for that, of course.
Yes.
And they're probably doing it at the moment, but
well, if they can't get the tissue though.
Yeah, I don't know what the story is there.
So, Vincent, the solution here is obvious. You need to move your lab to Sweden.
Sure.
Yeah.
You know, I could live there. You know what the only problem is? In the winter, it gets dark at 3 p.m. That would be tough.
Yeah.
And it's getting chilly already. I mean, well, here's the thing. It never goes over 23, 24, or very rarely Celsius. So, kind of like the world.
It was 28 in one of the days I was in Norway.
They probably said it was really hot, right?
It was hot. I mean, that 28 is 82.
Yeah.
Yeah, but it gets even hotter here, right?
Yeah.
But that's probably rare,
right
right?
now.
Yeah.
Anyway, I could, but I'm not going to do these experiments. We'll leave it for other people.
Back on what you were just saying about, because you don't know what cell type it is. I liked this line where they said the human lung contains up to 40 different cell types.
That's amazing. Does that include immune cell types, you think, or just lung cell types?
I would bet it includes immune cell types, at least like an alveolar macrophage.
I don't know. They just says, including epithelial, endothelial, mesenchymal, and smooth muscle cells. So they don't even call out the immune cells that are there. But
So Brianne, how many immune cell types would we find? We got T and B cells. We got macrophages, dendritic cells, NK cells.
Yep, neutrophils, eosinophils, basophils.
Up to 8.
Mast cells.
Mast cells. 9.
Yeah.
So most of them are structural lung cells of some kind, right? I guess, that includes the blood vessel cells, right?
Sure.
All these specialized epithelial, like the mucus secreting ones, the club cells. And, down in the lower parts of the lung, you have flatter cells that form the alveoli, right? And up at top, you have columnar of different kinds. Yeah, so I could see where there'd be 40. But that's a cool number. Yeah, I hadn't seen that before. Now you can remember. Hey, did you know that there are 40 different kinds of cells in the lung? One of those factoids. Okay, so there you go. That's a cool application. I hope that we're able to do this in the future.
They also point out that in addition to doing this with lungs, you could potentially do it with other organs.
Sure. Oh, yes. It's very important. Yes. Yeah, you could do it with liver. Well, I guess these mice do get a liver piece, right?
They do.
So is some part of the liver human, or is that just helping to establish the immune cell transplant? Does anyone know?
I don't know if that's a functional piece of liver, or if it's just other than the immune part of it.
I'm not sure what they transplant.
It's definitely human liver and thymus.
Yeah.
Right.
So
the thymus, they say, allows for human T-cell education, right? Does the liver do that as well?
It does not. I know the liver usually contains a fair number of NK cells, but I don't know if it's required for this transplant.
I wonder if you could put brain pieces into...
I was just thinking about that. Yeah.
Probably.
Yeah.
Get little mini brains. You could do kidney. So you could do intestine.
You could probably do anything where you could also do an organoid.
Yeah,
right.
Yeah.
Mouse.
You'd have to sterilize it.
Yes, you would have to. I wonder if you put an organoid in, whether it would further differentiate because it's got a nice environment, right?
Yeah.
Right. Yeah.
So that may get around because there are lung organoids for sure, right?
Right.
So maybe you could put those in and they go, oh, finally we're in a good environment, make up things or something. So that could get around the fetal tissue requirement, right?
It could. Well, it also could, allow other things like the immune cells to interact with the organoid.
Right.
Yeah. Yeah, I think it's important to get around this. And if people are wondering, you don't have to do an abortion to get this on purpose, right? These are tissues that would normally be discarded.
Right.
Yes.
If you're worried about that, that's not an issue.
Right. The other thing that was interesting when they listed the other lung human pathogens, they said enterovirus D68, adenovirus type 7. Why did they pick type 7?
Type 7.
Why not almost all of the lung adenoviruses? Like type 14 is, has some pretty virulent types right now. I don't know. I just thought that was a little bit odd.
Well, there's probably not a single adenovirus person on the paper, right? So that if you had been on the paper, then you would not have said let them say that. Yeah.
Adenoviruses.
If you had provided some adenovirus for them.
It's true. There are adenoviruses that infect the intestine and the eye, but the vast majority are respiratory that we know about.
Yeah.
Anyway.
All right. Let's do some email. The first one is from Sophia. Now, Sophia came up to me at the meeting in Smögen and said, "I
love saying that."
Well, look, they taught me how to pronounce it because I said Smogen, right?
Right.
And I went to the hotel and checked in. The lady said, "Where are you going?" I said, "Smogen." She looked at me like, "What?" And then she looked at me and I said, "That
There's
island."
No Smogen in this hotel.
You mean Smögen?
Smögen.
So, and in fact, I went before the podcast. I went onto my computer and just did a pronounce thing, right, where you can play. And even within Sweden, there are different ways of pronouncing. And you have, so that's an O with 2 dots, and then you have an A alone, you have an A with a circle, and you have an A with 2 dots, and they all have slightly different sounds. So it's pretty interesting to do all that, but that's why I'm saying Smögen, because there are some people in Sweden listening, including Sophia, and I want them to know that I remember how to say it. But I only know 3 words in Swedish, so I know tack, hey, which I love because it's hello, right? Hey, hey. Yeah. So I always say hey, but they do hey or tack. Okay. Anyway, Sophia came up. She said, I sent you a letter a while ago. I said, I didn't get it. And so she sent it right there. So here it is. This email is sent from the beautiful capital of Sweden, Stockholm. It is way overdue, but better late than never. I think she sent it in, July. Some time ago, a coworker of mine here at the Public Health Agency of Sweden asked me, "Do you know this podcast called This Week in Virology? It's really good." I looked at her and smiled. "Do I know of TWIV? I personally know 2 of the people in the podcast, Rich and Alan." I could tell she was super impressed. I then forgot about the conversation until I went to a meeting last year, the Smögen Summer Symposium on Virology. I think you know which meeting I'm talking about, but for them who don't, every year the Swedish Society for Virology have a meeting on a small island called Smögen on the west coast of Sweden. It's a really good meeting with both national and international participants, great science, and the best seafood ever. Well, I can't really back up the seafood statement since I'm a vegetarian, but the meat fish eaters seem happy. Anyhow, at the end of the session about vaccines, the speaker put up a picture of TWIV episode 496 and said, "If you want to hear more about vaccines, you should listen to this really good podcast and their vaccine episode. It's about 40 minutes long." I could not be quiet and raised my hand. You're absolutely right. It's a great podcast, but 40 minutes? Come on, they haven't even finished their weather reports in 40 minutes. After that, the majority of the audience started to talk about your pod. It turned out that most of the people in the room knew of or listened to TWIV, so you don't just have one fan, me, in Sweden, but many. How I know Rich and Alan: I did my PhD at the University of Florida and met Alan through Dr. Condit, Rich suggested I invite Alan as a speaker for our career seminar series. Each month we invite a speaker with a PhD in biomedical sciences to talk about their careers, purpose to expose us, the students, to other options other than the academic professor route. Alan spoke about being a science writer, and his talk was very appreciated, mostly because it was an interesting talk, but also because he was honest with the pros and cons of the job as a science writer. So thank you again, Alan, for your seminar and advice.
Thank you for inviting me.
As a microbiology immunology grad student at UF, Dr. Condit was one of my teachers. One of the first encounters I had with him was when I had just gotten the smallpox vaccination because I rotated in a lab working with vaccinia. Dr. Condit was very excited about this. Me, not so much, since I got a bad scar from it. Even if I did not share Rich's enthusiasm for that particular vaccination, I appreciated all other discussions we had. Dr. Condit's passion for science and virology always shined through, which was very refreshing and inspiring. I don't know if I ever thanked him for that. If not, thank you, Rich, and thank you for coming to my defense, even though I chose HIV over pox viruses. As a matter of fact, not too long ago, I actually got proof that Dr. Condit has inspired others too. I was discussing viral vectors for vaccine delivery with one of our English collaborators, and pox viruses came up. I know a pox guy, Rich Condit. My colleague looked at me. You know Rich Condit? I explained how I know Rich and asked how he knows him. Oh, I don't know Dr. Condit. I only know about him and his work. Very impressive and inspiring. Now my colleague asks about Rich every time we meet. So in conclusion, the podcast and you are all very appreciated amongst virologists, immunologists in Sweden and England, and I hope to see as many of you as possible in Smögen this year. It's time for a second road trip in Sweden. Rich, cheers. Sophia, PS 4 p.m., 17 degrees Celsius outside. Not the best Swedish summer day, but it will be better in August, I promise. It was very nice. Except in Smögen, it rained the first 2 days. But then it did clear up, and Friday night we went on a boat trip in the archipelago, and it was cool, and they had big platters of shrimp and ocean prawns there. It was very good, very nice. It was lovely. The Swedes are very friendly. I love all of you. You're great. Who didn't take one earlier? Kathy, did you not take?
I did take one earlier.
Brianne,
did one. Did Brianne?
I did not.
Go ahead.
All right. Neva writes, "Hi, gang. Thanks, Rich, for channeling my thoughts about the Ars Technica article and TED Talk. Get a load of this bozo about covers it." I remember a few weeks ago, Rich was talking about this TED Talk.
Yeah.
I thought that the old ferret arguments had settled down, but instead seemed to be entrenched. Okay, insufficient reporting again. Reed's favorite fantasy scenario of some lab creating a truly terrible disease as an experiment or weapon is, however, one that people could be rightly concerned about. Reed also believes, as he said, synthetic biology could be the greatest boon to humanity in all of history. He has good ideas and some missing info and could profitably spend some time talking to a greater variety of scientists to lower the BS level. Great podcast as usual, gang. Many thanks.
So in one of Erling Norby's books, he wrote, quote, "There is no reason to forbid any form of science, but it is important to ensure that there are ethical rules which guarantee that new information is used and not misused." That's why I agree with that. The problem is the public often doesn't think we can have the right ethical guidelines.
Yeah. Right, exactly. Well, that's why we have to show them that scientists are just normal people.
We try.
And synthetic biology, by the way, is biochemistry plus marketing.
Yep.
Yeah.
So one of my guests at the Karolinska had a cool definition about systems biology from Sidney Brenner. I can't remember, but when you listen to that, you'll hear. It's from someone you must know of, Brianne, Peter Brodin.
Yes, I do know of him.
Yeah, he was one of the three guests, and he had this funny quote from, because I asked him, because he does what he calls systems immunology, and I said, is that like systems biology, physiology with marketing? And he said, no, it's even better. Here's what Sidney Brenner thought. Very cool.
Well, I look forward to listening to that then.
Alan, can you take the next one?
Sure. Jolene writes, "Hello, Twivsters. From the discussion of measles virus budding through the apical surface of lung epithelia, I can't help but wonder about the biophysics of that virion release. When those particles, or any virus, shed through the airway are pinched off, it's not like they are now floating in a bubble or aerosol droplet on their own, but in a layer of mucus, right? I believe I've read that the deepest recesses of the alveoli are warm, wet places. I imagine that the force of coughing could dislodge mucus with the virions in what become aerosol droplets of varying sizes." That's a good point. So it buds out of the cell, and then it's sitting there in a vesicle in its envelope in mucus.
Mm-hmm.
But then the mucus gets aerosolized to come out of the lung. Would be the next part of that mechanism, I guess.
Well, this is a good point because a lot of transmission occurs just by talking, right?
Right.
Because you make big aerosols when you talk, and especially with the P's, right? And those,
right.
What do you call those letters that make
fricatives?
Floatives.
Is that what
plosives?
Plosives.
Right. There's plosives and sibilants and plosives. So not all of it's coming down from the lower lung, right? So I guess if it is coughing would be a big part of that, but I think a lot of it's from upper lung, which can also be, the upper lung parts can also be infected. But people have worked on this extensively. I'm just talking out of my memory. I'm sure I'm saying something wrong here, but
memory was not the word I thought you were going to use.
Yes. In fact, there is a past TWIV episode about using a machine to quantify that. It's one that's a choice for a student assignment, and it seems very popular. So I read a lot about that episode every spring.
Yeah, that's the one with a picture of a guy who looks like Alan.
Right, he's got his face in a traffic cone.
Yes, and they built a machine to measure sneezes.
What do you call that machine? Oh, there's a great name for that machine.
Name.
Oh, yeah.
Yes, so you put it and you just talk. They have you go through a lot of words with different properties.
Right.
And
then they measure the aerosols coming out of your lung.
So for flu, it turns out that most of the transmission is just by normal talking, not so much by sneezing and coughing. Don't have to. In fact, I think coughing was the least one that gave virus on the traffic cone. That's right. It did look like a traffic. And by the way, a lot of that happens at the University of Maryland, which is where we were last year. And I remember encountering someone who was involved with those studies. So, there is. There's mucus coating the entire tract, right, from the top to the bottom.
And the episode is TWIV 480, and the machine is the Gesundheit 2.
Gesundheit 2, that's it.
Gesundheit 2, right? Gesundheit, right. So, this is an interesting question. It's going to be in the mucus originally, but somehow it gets out, and I don't know how that works. So someone must know, or else we will look it up and tell you next time.
It talks its way out.
Kathy, can you take that last one? If only because you will pronounce the name correctly.
You think it's Trolls, right?
Yeah.
Dear TWIVologists, thanks for a great discussion of the hepatitis delta paper in TWIV 552. To me, one of the most exciting papers this summer. Fascinating that HDV can utilize not only HBV, but also other viral envelopes to package its genome. It will be interesting to follow what real-life examples will be found in the future. As a virologist passionate about hepatitis viruses, I couldn't help stumbling upon your alphabet list of hepatitis A through G. Someone, Rich Condit, saved it a bit by explaining how hepatitis F was a fluke. Unfortunately, here, like many other places, G was still mentioned as if it was a hepatitis virus. The course book for medical students at our university is no better. In the excitement over identifying novel hepatitis viruses in the 90s, conclusions were perhaps drawn a bit prematurely, and unfortunately the name sticks. It happens to be that HGV is not liver-specific. It does not cause hepatitis, and most evidence suggests that it doesn't even replicate in the liver. Rather, it is a rather interesting virus that flies under the radar and can cause persistent infection with no clinical disease belonging to the genus Pegivirus within the Flaviviridae family. So let me read that again. Rather, it is a rather interesting virus that flies under the radar and can cause persistent infection with no clinical disease belonging to the genus Pegivirus within the Flavivirus family. when discovered in parallel in two different studies. One study termed the virus HGV, while the other termed it GBVC. Shortly before that, two other viruses of the Flaviviridae family had been discovered after 11 passages in tamarins of an acute phase serum during hepatitis from a surgeon with initials GB. These were termed GB virus A and B, although to date it remains unclear whether they were ever in the original human sample. Because of phylogenetic relationships, in particular to GBVA, and although completely unrelated to the GB sample, the new human virus was named GBVC. To avoid confusion, and perhaps because the two original names were equally inaccurate, it was recently renamed human pegivirus HPGV. Whew. Okay.
Ooh.
In the same episode, you end up talking about horses and liver disease.
Note to self, never mention hepatitis G virus again.
Yes, please.
Whoa.
Yes.
Yes. Okay, the same episode, you end up talking about horses and liver disease, and there I couldn't help noticing a very convincing reply from Vincent when the question came to whether hepatitis viruses in horses are known. Not too well known, I would say, because we are really just beginning to understand what's going on there. Despite infectious liver disease in horses, Tyler's disease has been known for 100 years. Infection of horses with the closest relative of HCV, non-primate hepacivirus or equine hepacivirus, NPHV or equine HV, EQHV, discovered by Amit Kapoor in 2011, indeed resembles HCV infection a lot. It is a liver-specific virus that causes hepatitis as defined by infiltrating lymphocytes. See, for example, a paper in PNAS. But perhaps not much more than that. In contrast to HCV, most NPHV infections in horses do not become persistent and are not linked to any clinical disease. And he gives another reference. The question is whether the lower fraction of chronic infections can cause liver disease in horses, and case examples begin to suggest that it may. In another story, strikingly similar to that of HGV, another PEGI virus, Tyler's disease-associated virus, TDAV, was linked to an outbreak of acute serum hepatitis after administration of botulinum antitoxin in horses in Nevada in 2013. And he gives a reference. Biologicals given to horses are often produced in horses without subsequent purification. Oh. Given what I mentioned above, that PEGI viruses normally are not associated with disease, this came as a surprise. Further, no other TDAV cases could be since identified in horses with liver disease. Surprisingly, collaborators of ours, Tom Divers and Gerlinde van de Waal at Cornell and Amit Kapoor at Nationwide Children's, instead found a novel parvovirus in almost all horses with unexplained infectious liver disease tested. And he gives a reference for that. Looking back at the Nevada outbreak, it turned out that all the affected horses and the responsible lot of antitoxin was positive for both viruses, TDAV and the parvovirus. It just happened to be that viral discovery had only been done on RNA, not DNA. Together, we are currently finishing a study on TDAV showing that this equine pegivirus does not lead to clinical symptoms, and evidence for negative strand RNA replication is exclusively found in the bone marrow. This tropism happens to align well with a recent study on simian pegivirus, and he gives a reference for that, and older case reports on GBVC. So, for humans, we can agree on hepatitis A, B, C, and E. I belong to those who wouldn't consider Delta a real virus, but that's a completely different discussion. For horses, we're probably looking at a parvovirus, perhaps EQHV/NPHV, and who knows which others. And yes, one of the two microRNA 122 inhibitors tested in clinical trials for HCV indeed is pegylated. This does increase its stability, and it is nothing less than fascinating that several patients obtained virological cure by a single shot of this inhibitor. He gives a reference for that. A disease that just a few years back could often not be treated despite year-long therapy, with severe side effects. Thanks for a great show, and looking forward to seeing you next week in Smögen. Charles. P.S. Sorry for the length of this email. It was my intention to keep it short.
We feel the same way about TWIV.
And he is an associate professor at the Copenhagen Hepatitis C program at the University of Copenhagen.
Cool.
Huh. Did we meet? I don't remember.
He might not have introduced himself to you.
Maybe not. Well, if you're in Copenhagen...
there's another chance.
Another chance. And if you're going to the meeting of the European Society for Clinical Virology, which you might or might not, trolls, come by. This is all very good stuff.
Yeah.
So let me just make a few comments. First of all, you listen really with great attention to detail. It's impressive.
Yes.
I don't know if you do that for every episode or just the ones you're interested in, but it's pretty cool. Yes, hepatitis G. I am also guilty of including it in my list of alphabet hepatitis, but I will leave it out from now on because, yes, I know that story. It's been recounted before. I think we had Amit. Yes, of course, we had Amit Kapoor and Ian Lipkin, and I'm sure they talked about it for sure. Absolutely. But our memory is short. And, once hepatitis G is in the literature, it's really hard to get rid of it.
And it's hard to get it out of your mind. I mean, if you learn something in undergrad and were tested on it, it just sticks.
But I'm looking at the Viral Zone page for PEGI virus, and I don't know if this is a holdover, but under tropism, it includes liver, spleen, and peripheral blood mononuclear cells. So maybe they need to fix that too.
Yeah. So
that's one thing. The other, yes, he said, Vincent was very sure about hepatitis in horses. Well, yeah, I was sure, but I was wrong. As you say, I was thinking of the Hep C virus, which seems to have infected horses, as you say, but it doesn't cause any clinical disease. So thank you for that. And the last thing, oh, yes. Of course, deltas are virus.
Yeah.
Viroids are viruses. The virus doesn't have to have its own capsid. I think it just needs to have other properties getting into cells and being a parasite and so forth. So anyway, go ahead.
I was just going to say, and I still think of it as the horseshoe crab of viruses.
Yeah, that's a good way to put it. It certainly is. And there are certainly others out there. But anyway, that's a great email. Thank you. Thank you so much. And keep on listening. Let's do some picks. Brianne, what do you have for us?
I have a paper that was in Nature Immunology recently. It was also all over Twitter. This was written by Akiko Iwasaki, who was the keynote speaker at ASV this year. And in fact, because so many people were asking questions on Twitter, I know for a fact that it's open access because she made sure that it was.
Yep.
It's called Why We Need to Increase Diversity in the Immunology Research Community. And Akiko does a really great job of talking about all of the benefits to science that we see when we have diverse research groups and diverse researchers. I think that most of the things that she talks about are just as applicable to the virology research community as the immunology research community. And I think her recommendations will be really helpful as well. I liked some of the things that she talked about with regard to leadership and different ways to think about what a leader is.
I think it's probably applicable to all fields, right? Especially science fields for sure. That's what we can talk about.
Yes.
I figured she limited it to immunology because, hey, diversity.
Diversity.
I know. The beginning is really nice because she says, well, immunologists obviously know that diversity is important. After all, we think about all sorts of aspects of diversity in the immune system. And so it is sort of a nice beginning there.
Mm. Alan, what do you have?
I have something I have been hoping somebody would do for a long, long time. Every time I see a story about vaccination, or 9 times out of 10, if they illustrate it with a photo and everybody's seen this, it's somebody sticking a huge needle in the arm of a screaming child, right? I mean, you've all seen this. Get your flu shot, and wah! That's the illustration for it. And that is not helping. It's medically inaccurate. It's just playing on people's fears. And so Self Magazine, and a huge, huge congratulations to them, because this is a tremendous public service. They have put up a gallery from a photo shoot that they did. And this is, they got a group of people together, including pediatricians from the AAP. They had obviously professional photographers and their models all lined up. And if you follow the link, you can find this entire library of images either on Flickr or from the American Association of Pediatrics website. And they did a great photo shoot. These are good photos, just photographically. The lighting is good. The poses are good. They're medically accurate. You've got appropriately sized needles in the cases that needles are being shown. They're ethnically and body type and every other kind of diverse. So you've got a population of people, folks can look at the picture and see themselves getting a vaccine. And just this is just all kinds of great. And they're Creative Commons licensed. So
nice.
if you're all you editors out there, if you're doing a story on vaccines, you go here, you download the image. All you have to do is credit the source, and it's free to use.
Mm-hmm.
Plus, there's a link to another page called Vaccine Saves Lives.
Yes.
It's a project where they have various articles which are great. Like, I have a compromised immune system. I rely on herd immunity to live. And she tells her story. Mm-hmm. And there was another one. I used to be an anti-vaxxer. Here's what's changed my mind.
Yeah.
A crunchy mother of 6 shares her story.
Yeah.
This is great. Thank you. I'm glad they're doing that.
Yeah.
I wish more photos are awesome.
I wish more major mainstream publications would do this.
Yeah.
Right?
Mm-hmm.
Very cool.
And they can now. That's because the photos are freely available. So no excuses.
Hope we see these photos all the time.
Yes.
Kathy, what do you have?
Well, as I mentioned, I was in Norway and I was visiting with Wendy Mari, who you may recall was on the episode that we did from Iowa in March. And we were talking about sample sizes and numbers of animals. And she said she had found this website that has a sample size calculator. And it's a really good one because you can put in various things and you can see, if your mean is 30 for one treatment and 20 for another, and you want a certain power, then how many animals you have to use, and then you can see how it changes. If the means become closer together, you need more animals, and so forth. And so it's just a really convenient, easy site, and it explains, the type 1 and the type 2 errors and all the component parts of the calculator. So it's really helpful.
This is nice.
This is great, but
this is awesome.
I can't use it for IACUC because they'll say, what's the calculation you use? I can't say I use this website.
No, but I also have I can send you the reference that I have for that. That's basically the same thing, but this does it for you electronically.
Automated. Yeah. If you know the formula behind it, that's good.
Yeah, you can actually, you click on the button, view power calculations. I just did that. And it gives you this wall of mathematical stuff that I'm sure is going to satisfy any IACUC.
Yeah, I want to give them a wall.
It and say, oh, they did math. Okay.
Yep. Yeah. I want to give them a wall because they asked for it. They're getting a wall of
just
math.
Click view power calculations and paste that in.
You know what kills me about this? I've been doing this for 30 years, using 5 mice for this, and then just in the last year, please justify why you're using 5 mice for these experiments. No memory whatsoever. Of course not, because they're all new people. And they just want to generate a paper trail. Yeah. That's very cool. Thank you, Wendy. That's great. Very useful. My pick is a book this week, which is called How Economics Shapes Science by Paula Steffen. So Paula is a economics professor. Oh, is it Georgia State or Georgia Tech?
Oh, they're quite different.
I know. Paula, I have to look it up because people will get mad.
Georgia State University.
Okay. And this book is about, as it says, the economics of science, but it is just great. She is not a scientist. She's an economist, but she gets it all right. And, the first half of the book is explaining how research is done, mostly in the U.S., how it's funded, what fraction of, government funding versus other funding, and then how research is done, and then the economic impact. And it's great. And the first thing I thought after reading this is if I had read this before I went to grad school, I wouldn't have gone to grad school.
Yeah.
because it's pretty sobering. And she goes into things like, the effect of the NIH budget doubling, how it screwed up basically the whole situation. You want to know, read it. She explains it beautifully. She talks about the pyramid that science research is.
Yeah.
And, how investigators use postdocs and PhD students to do their work, even though they're not jobs for them. And she talks to some investigators who say, it's okay. They know that. In the U.S., so she gives you the numbers for exactly how much science drives the economy. It's quite interesting in how much it makes you live longer and so forth. And then there's a very interesting statistic that most of the research in the U.S., I would say the majority of research is done by immigrants. And why? And she goes through a lot of, and you probably know this, Kathy. I think most of the faculty at the University of Michigan, at least the majority, has come from a different country.
In our department, that we have a large number of international people.
Yeah. I think it's a lot also in things like engineering and physics and so forth.
Yeah, probably.
But so the number of people coming. So it's important to maintain that base because they do a lot of the research, and research drives our economy. And she says the problem is that base is decreasing because of ridiculous immigration rules that are being put into place. So she said that's an economic problem that has to be recognized. Anyway, so that's just part of why I really like it. It's a few years out of date, so the numbers aren't all right on, but it's really, really, she does a great job. Great writer and really explains everything really well. So how economics shapes science. Very good. We have a listener pick from Melinda. I have been using Zome tools to build viruses in my classes. They are great for building icosahedral shapes. Zometool.com/shop. One picture shows all my 3D printed viruses. She sent a couple of really nice pictures. So these are 3D printed viruses. You can go right now, Brianne, just get out your credit card.
Perfect.
And shop.
So they're like Tinker Toys for scientists.
Mm-hmm.
These are cool.
Yeah. Oh, well, they're 3D printed things of all sorts, right?
Right, but
you can put them together. Yeah.
Kits of struts and connections, and you can put things together in multiple ways. And cool.
Thanks for all the Twix podcasts. Melinda is a professor at the University of Georgia. Thank you, Melinda. And that it will do it for TWIV 563. Microbe.tv/TWIV. Questions and comments, TWIV@microbe.tv. Please support us. Microbe.tv/contribute. You can go to PayPal or Patreon. Give us $1 a month. Helps to support our habits here at the Microbe TV.
And by the way,
podcasts.
Often when Vincent is talking about travel, for example, to Sweden, that's not been paid for by TWIV budget. That's a lot of that, I understand, is paid for by the hosts of those shows. So,
yes. So that's the other thing. When I travel, usually people invite and they pay me, but not anybody else. And I would like to be able to bring someone else. Sometimes I ask and they do support travel. Like for TWIV 500, we were all there last summer, right?
Yeah. That was good.
Or, if Rutgers invites us, that doesn't cost anything because we drive. Right. And Dixon and I and Brianne, we just drive down the road. But if we want to go somewhere else, would love to be able to bring the teams or part of the teams that are, and as you know, travel is expensive. But even if you can contribute, that would help. Microbe.tv/contribute. Brianne, yes.
Oh, I was just going to say another connection in these pics. Melinda was a graduate student of Wendy Mari's.
Now, did you know this? You just looked it up.
I confirmed it. Wendy had mentioned it the other day, and I just wanted to confirm that this was the right Melinda. Yeah.
Now, how did she know to confirm it? Because you said we have this Melinda person here that has a letter. Do you know her? Or
No, I have met Melinda myself, and I just, of course, PubMed just put in Brindley and Mari to make sure it's the same one.
Okay. Yeah. Cool. Brianne Barker is over at Drew University on Twitter. She is BioProf Barker. Thanks, Brianne.
Thanks. It was great to be here. I'll take a break from my shopping to say thanks.
What's the course you were teaching this fall?
I am teaching a first-year seminar on HIV and an immunology course with a lab.
All right.
Wow.
Have fun.
Yes, we are having a lot of fun.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was a lot of fun.
Alan Dove is at turbidplaque.com on Twitter. He is Alan Dove. Thanks, Alan.
Thanks. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank ASV and ASM for their support of TWIV. Ronald Jenkins for the music. This episode of TWIV was recorded, edited, and posted by me. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWIV is viral.